The blood-brain barrier is one of the body's most selective gatekeepers. It separates the circulating blood from the brain's extracellular fluid with remarkable specificity, allowing in what the brain needs and blocking most of what it doesn't. From a supplementation standpoint, this barrier is critical. A compound that cannot meaningfully cross it cannot directly influence brain NAD+ levels, regardless of how well it raises NAD+ in other tissues. NMN marketing frequently implies superior brain penetration and neuroprotection compared to older NAD+ precursors. It's a compelling claim. Who wouldn't want better brain NAD+ delivery? The problem is that the evidence underpinning this claim is substantially weaker than the marketing confidence suggests. Multiple studies indicate that NMN undergoes significant degradation before reaching the blood-brain barrier, being cleaved into smaller components by enzymes along the way. Whether meaningful concentrations of intact NMN are available for brain uptake in humans remains genuinely unclear. Nicotinamide, the metabolite shared by multiple NAD+ pathways and closely related to niacin, crosses the blood-brain barrier relatively readily through specialized transport mechanisms that have been characterized in detail. This means that niacin, metabolized in part to nicotinamide, generates a compound with established brain penetration capability. The irony is that the supposedly inferior, cheaper compound may be delivering its active metabolites to the brain more reliably than the premium molecule being marketed specifically for its neurological benefits. Niacin's flush mechanism adds another brain-relevant dimension. The vasodilation triggered by prostaglandin release increases cerebral blood flow. Better cerebral circulation means improved delivery of oxygen, glucose, and other nutrients to brain tissue, a benefit that operates through an entirely different mechanism from NAD+ synthesis. Someone taking niacin for cognitive support may be receiving both NAD+ related benefits through nicotinamide conversion and hemodynamic benefits through the flush mechanism, while an NMN user receives neither of the vascular effects. Individual variability complicates this picture considerably. Genetic variants affecting NAD+ transport proteins, blood-brain barrier transporter expression, and local NAD+ synthesis capacity in brain tissue all influence how different people respond to different precursors. This is why blanket claims of superiority in either direction are scientifically premature. The appropriate response to this variability is not to assume the most expensive option is correct, but to acknowledge that individual testing and tracking of cognitive outcomes provides more useful information than any preclinical study or marketing document. A structured self-experiment, baseline cognitive assessment, supplementation period, washout, reassessment is a more honest way to determine what works for a specific individual than trusting marketing about barrier penetration.